Uh, it's going to be a really interesting talk today. We have Daniel Díez from the University of Basel. And he's going to talk about Dynamo, which is a package for tomography and subtomogram averaging in particular. And it's probably going to be the next big area in cryoEM, in my opinion. I think this is going to be the new frontier, and this is going to be where a lot of the interest and developments are going to come in the next couple of years. So it really is an opportunity to see kind of where the field of cryoEM as a whole is going to go and where a lot of the new techniques are going to arise. So with that, I'll just ask Wolfram to change over and remove his intro slide, and we can get Daniel sharing.
I hope you're seeing my screen now.
Yeah, that looks good. So I guess all there is to say is take it away. And if anyone has any questions, just feel free to ask them in the chat. Thank you very much, Daniel.
Okay, then let's start. So as Shaun explained, Dynamo is or was at the very beginning a software package for subtomogram averaging. But what I want to present now is some of the extensions that we have been developing in the last years to make it suitable for the full cycle of cryo-electron tomography. So that I will talk about subtomogram averaging itself, also about particle picking for subtomogram averaging, and about tilt series alignment. And also, as I'm talking for SB Grid today, I will talk also a little bit about Dynamo as a development system and what is inside Dynamo itself. So that let's recapitulate what would be the standard pipeline when you are doing subtomogram averaging or in general cryo-electron tomography from the computational point of view. You would start with a tilt series. Well, actually, you would start with a lot of tilt series, but let's play with me for this simplification. That you have to align. The aligned tilt series is still a stack of bidimensional images related by geometrical transforms that you have to find, or that have been already defined in the previous step. But you still need to create a reconstruction in 3 dimensions, what we call the tomogram. In the tomogram, we need to locate the particles of interest, which we call the subtomograms. And once we have access to them, we align, average, classify them, and produce our density map or family of density maps. That's what I call the classical pipeline for subtomogram averaging. In the last few years, new development is arising. We are living exciting times in subtomogram averaging, in which the average found in the, let's say, classical pipeline is used again in order to feed the information that you had when you were aligning your tilt series and cycle again and get better and better resolution. That's what you can do with EM clarity. That's what you can do with M from Dmitry Tebunov, and that's what you will be able to do also with new releases of Dynamo incoming. Also with EMAN too. Now this is more or less, this scheme that I'm showing now is becoming the new standard in subtomogram averaging. In any case, if we go a little bit back, I want to explain the different modules of Dynamo in a separate fashion, and I will start chronologically. Actually, this is the first module that I wrote, in which we assume that our subtomograms have been already located in our tomograms, and is just a question of align and average them. This module was written in Basel just for the special requirements of people in my lab or in the lab of Henning Stahlberg, which found that the typical STA pipeline for membrane proteins was not working well with our sample. So that in principle we started small with this system. And immediately the users in my own lab started to ask for, well, your code is not bad, but I would need it more user-friendly, I need it faster, I need it more flexible. And following those guidelines of my users, first we created a pipeline for basic users, people that don't want to dedicate your life or to subtomogram averaging, you don't want to understand every possible parameter, but you just want to have a basic idea of what you're doing and have a good life, just inputting your data, setting parameters, running the projects. That's what Dynamo can also do for you. You can let it drive the ride with optimized parameters using a golden standard-based adaptive filtering. And that will do for many users. For some other users, in Dynamo, we have a very extensive toolbox for a big variety of tasks that typically, or not so typically, but sometimes appear when you are working your way in a STA project. And I would say later why this big variety of tools in the toolbox is necessary. Some of these tools are, well, not so spectacular, but they are really necessary when you are in the specific case in which you need it. For instance, something as simple as tools for a specific 3D drawing in the typical gray map representation of averages of particles. That's something that you can find in the typical graphic design software, but this is adapted to the requests of subtomogram averaging, or how to depict the relative positions of your particles and constitutive elements on your tomograms, or sometimes models that are much more complicated, like the different ways of running classification tasks in Dynamo. In any case, one of the basic ideas on this module in the STA, in the subtomogram averaging per se module, is the GPU computing. Just want to say very quickly here that when you are creating algorithms for the GPU, the thing that you have to take into account is that the major constraint that GPU computing is imposing onto you is that the GPU is designed for performing many operations on one data unit. If you do it in any other way, you are going to suffer because transferring memory from the host to the device, to the GPU, takes a toll. So that basically you want to put data in the memory of your device and then a lot of things, then do a lot of things with this piece of data inside your GPU. And that is almost a verbatim description of what is subtomogram averaging. Once you have in your GPU your template and your particle, all you do is you oppose to the particle or to the template, depending on the implementation, thousands, hundreds of thousands, millions of rotations and three-dimensional FFTs. My experience, normally the FFTs are the thing that you have to time. At the end of the day, the FFT goes as n log n, main n the number of pixels. That's why the subtomogram averaging is a perfect scenario for getting a nice speed up through GPUs. Those are some numbers I got a couple of years ago. These GPUs are a little bit old. I'm really looking forward to put my hands on the 3090 and the DGX of NVIDIA, but I expect to have even better speed ups with the new devices. Also, I also looking forward for Amazon EC2 to offer new instances of GPUs, because we offer in Dynamo an AMI, basically a pre-configured virtual environment, so that if you don't want through the pain of installing things, or if you don't want to go through the pain of deciding, do I need a GPU for this or not? You just can open an account in Amazon EC2 and access our EMI, and yeah, there is a small but actually I would say that computation time is quite cheap in Amazon EC2 even for the GPUs. Storage is not so much, so that be careful how much you play and how long you play with your data in Amazon. In any case, that was our first module in Dynamo, and we were happy with that. It was good enough for our internal users in CINA, in Basel, in the Stalber group, so that we say, okay, let's train external users in tomography. And since 2012, we do our yearly Dynamo workshops in Basel. We also participate in third-party workshops like the EMBO courses for image processing, the CCPM courses. Sorry. And that's important for us because that gives us a very robust, very important feedback about which tools are actually needed by the subtomo community, a community that is actually increasing in the last years. And we always expected that the wish list will be something like that. We actually, after the workshop, give the participants a formula, please. What would you like to see in Dynamo? And we always expect to see things that, okay, I want to have better resolution, faster code, better classification schemes, better CTF, etc., etc. That's what we expected. In reality, what we almost systematically see in our feedback forms is that what people really want and the moment in which people suffer the most is here in the particle picking part stage of the procedure. This is what we actually always see for two reasons. One of the reasons is that this small red box, that I showed before, where you have a tomogram and then the red spots that represent the particles magically appear, it was an enormous simplification. That's not how things happen in reality. You have lots of tomograms, and the way or the path between the tomogram and the individual positions and orientations, if possible, of the different particles is not always trivial. That is one thing. And the other thing is that keeping track of everything that is happening, it's something that is frequently underestimated at the very beginning by new practitioners of the technique. The data management overhead, just the data management can be really important. That is one thing. The other thing is that tomography inherently can present very different phases. Your particles may be distributed in very different fashions, and it's difficult to establish automated pipelines in order to just access your field series, create a tomogram, and inside the tomogram just extract everything that is meaningful for your project. Nowadays, there are a lot of advances in this direction towards an automated segmentation of tomograms, but we are still not there. So that still these days, I would say that each new tomography project is very much a new story for new practitioners. So that in our second module for tomography, what we suggest, what we present is this catalog system, which is kind of a database that takes for the user most of the effort of designing a rational way of organizing your data. We try to make the critical energy, the activation level of use of the catalog as low as possible. If the user sees that, oh, I really need to do a lot of things just in order to start to have a grasp of what is happening, people tend not to use it. People tend to create an Excel file that will become totally over complex in 3 or 4 weeks, and then it will be maybe a little bit too late to do things right from the beginning and create a repository or any other of the tools that we have in Dynamo to rationalize your work in tomography from the beginning. The things that you do in the catalog are, well, they come from the simple thing, like an easy way to visualize your different tomograms, to access them with a system of memory control, because remember that we are talking about tomograms. They can go to, I've written here 40 gigas, but sometimes they can even be larger than that. This amount of memory will crowd a lot of workstations. Modern workstation can take this, but still it will be a lot of time just to load it, so that being able to do a hot transfer of just a region of the tomo or the part of interest into memory, it's already something valuable. But the actual reason I would say that we decided to create the catalog is as a way to articulate what we call the models. The models are a kind of Dynamo metadata, Dynamo object. Let's describe them, telling what they do. They channel the user intervention on a tomogram. They offer different ways to the user to intervene and to interact with the computer representation of the tomogram in order to create different ways to access the particles in the tomogram. Let's see what it means. For instance, let's imagine that we are working with a particular type of model that is specialized in particles that are covering a surface. So the first functionality of this kind of model would be about visualization. In this particular case, what this model will offer you in this area would be, okay, when you initiate the model and you tell the model this is one layer, that will be the yellow puts that you see in the screen now. You mark where one layer in the membrane is, and then you let the model to decide where the layer is or where the membrane is in the subsequent layers. You have some parameters to decide how to do it, but the idea is that this should be kind of semi-automated. This is about the visualization part of the model. The second part of functionality are the tools, the geometry manipulation tools that are attached to what you have visualized. For instance, here, that would be the result of the visualization part. You have checked in different points in the membrane. Now you want to translate these points in the space into an actual surface. And in this surface, you want to pick particles. What do? Well, the model will give you a series of tools in which you can first create a smooth interpolant for those points in each 2D slice. Then you can create a triangulation, you can smooth the triangulation, you can define a distribution of tentative particles on that surface. You can provide them some normal, some directionality. Then you extract the particles, then you create an average, and then you are already in business for subtomogram averaging, because everything is inside Dynamo, and it formats these results in a way so that you can plug it into the next step of into the next module or into the catalog in a way so that you can initiate later your alignment project. Obviously, if you want to do this, if you want to really go to each small membrane in your data set and play with the parameters, is this surface good? Do we have artifacts that can take a long time? The idea is the following. Normally, you will have tens or hundreds of surfaces of interest in your data set. So the idea is that you take one or two membranes of interest, you struggle with them a little, you make your battle so that yeah, these parameters kind of work, so that you are satisfied with the distribution of particles that you get for one surface, and then you launch this workflow, this extraction workflow, to all the other vesicles in your data set, to all the other membranes of interest. So that was the second module I wanted to speak about today. There is a third module about the alignment of till series that is taking more and more importance in the latest time, because in general, when I started to do tomography, I would just patiently click on each goal bit in a till series to create one alignment. I will explain later what I'm meaning with that. But I would do a manual alignment of a till series. With modern subtomogram averaging, this thing is not possible any longer, because if you really want to align manually many till series in the scale that you suppose, and that is the standard today, in which you may easily have hundreds of till series, well, that's automation is really needed. You shouldn't align manually all this amount of till series. What is this alignment? What I'm talking about exactly? So imagine that this is a raw till series. You see the goal bits that should be nicely aligned, but that's not what the microscope gives us. What the microscope gives us is this bumpy transition between successive views of the sample, between successive tilt angles. So what can we do in order to correct for that? Here I'm doing a very small simplification. I'm ignoring the fact that the tilt axis has an additional angle. This is kind of trivial, that's why I'm not putting it in this equation. In principle, you would say, well, from the mathematical point of view, that is not so terrible. It's at the end of the day, we have some unknowns that are the shifts that are corrupting each one of the micrographs, so that if we could solve this least square problem, that is basically telling us, imagine that we have so many three-dimensional markers, how do we need to shift each one of the micrographs in order for our observations of the goal bits to match the expected projection point of each one of these three-dimensional markers. This is a least square problem. This is trivial, or this would be trivial if we had a clear idea of the identity of the trails of the fiducials that we see. Maybe that sounds a little bit vague now. Let me explain what I'm trying to say. Imagine that this is our simplification. This is what is happening in our tomogram. We have these colored markers with blue, magenta, and red edges, and the orange squares our micrographs. If I ask you to follow one marker, let's say the blue marker in the till series, it's kind of trivial, of course. You just see what is what. And that is more or less what you would do with iMod or Dynamo when you use them in manual mode. You say, oh, I see my goal bit in this slide, in this micrograph, and this goal bit is this goal bit in the next slide, and this goal bit is this goal bit in the next slide. That is pretty trivial when you're doing it manually. If you want to do it automatically, what you would do is, well, let's make a cross correlation against the template. We get some peaks. We have to threshold them to somehow, maybe it's not totally clear what is the best threshold. In any case, what I mean is that when you give your computer this, you can see that things are not as trivial for the human operator. What is what here? How do we identify which peaks of the cross correlation are actual goal bits? So that what we see is, well, let's say that the observations, and by observations I mean the cross correlation peaks, we have a trail of observation when it behaves like our actual projection should behave. But what is this should behave? Because at the end of the day, the alignment model itself depends on the position and identity of fiducials. At the end of the day, it's kind of a chicken and egg problem. The good thing is that in algorithmics, normally when you have a chicken and egg problem, it means that, well, try to refine iteratively. And that's exactly what we do in the automated tilt alignment module of Dynamo is this procedure in which, well, that would be the tilt series. We make our cross correlation for everything. For each micrograph, we locate the peaks. And then we try to locate which peaks are actual matches, meaning that when you move from one micrograph to the next one, we test all the possible shifts. And when we see that for this shift, we have a lot of goal bits that are matching each other from one micrograph to the next one, it means, well, they are probably the correct shifts. Or a good approximation. We do it for all the possible pairs of micrographs. And then we just postulate that as a seed model and we keep iterating. And that works at the end of the day quite well in our experience. This is what you want to see at the end of the day. Smooth transition. Oh, sorry. Well, that is what you want to see at the end of the day, a smooth transition from a projection to the next one. Another thing that I want to mention in this new module is that we also take into account that goal bits are not real spheres. We have to put some kind of thought into the fact that the goal bits will have different appearances during the tilt series. But in any case, again, this is the same idea as I mentioned before for the models for particle picking. The idea is that when you have a lot of tilt series, this is our naming convention in Dynamo for tilt series. Here I have 50 or something like that. The idea is that you choose one of them, you work a little bit with the alignment workflow, you tune the parameters in order to make it work decently. And if all these tilt series come from the same acquisition experiment, assuming that you have similar optics, you have similar distribution of goal bits, similar boundary conditions, you just use this workflow on everything that you have in your database of tilt series, and let it do automatically. It will always be some tilt series that will fail to align correctly with the parameters generic parameters that you found. The idea is that you can pick them one by one, or you can ignore them if they are few. And in our module, we include many new tools for graphical interaction with the tilt series in order to get you there manually in the case the automatic alignment didn't work. We have been using this alignment with basically everything we could put our hands on from NPR, and it's working quite robustly. We're deciding now what is the best metric to show these results in terms of RMS, percentage of goal bits that were actually located, etc. So that, as I said before, we are working into an integrative pipeline. We have databases for the different modules that I mentioned. In order to bring Dynamo to the next version, we need to integrate everything so that we can reuse the averages of classical subtomogram averaging for refining the till series, etc., etc. This is work in progress. And I just wanted to mention a couple of additional applications of Dynamo that we use it in the group of Henning Stahlberg for characterizing local texture in tomograms in which the segmentation proved difficult. Here, these samples, which are highly preserved human brain tissue, we were interested into checking what was the internal structure of subregions in the tomogram. So Dynamo has their modules in order to interact with the tomograms and create customized visualizations and use this visualization in order to pipeline into the STA workflow. And that's why how we were able to see in this subregion, the texture is more linear, is more fibril-like. In this other region, the texture is more membrane-like. And my last couple of slides. Many users are surprised when they found out that Dynamo is basically written in a big percentage in MATLAB. The reason for this design decision at the beginning is that, well, tomography is certainly not a streamlined technique like cryoEM is becoming. When you have a new idea, you have to do a lot of prototyping, you have to test a lot of different things. And to my taste, MATLAB, it's a very good environment for prototyping. And for users, I also think that tomography being, as I said, not streamlined, even if you are just an user that doesn't want to get very involved into the deep details of tomography, there are big chances that at some point in your project you will have to think a little bit out of the box, out of you have read, and you will need to do some kind of manipulation of your data that will require some customized task on your side. And I think MATLAB is something that gives casual user, let's say so, a very smooth learning curve and makes possible for people that normally would say, no, I cannot, I don't want to interact with this software, a good entry gate. There are some cons inherent to working with MATLAB, and in Dynamo we try to mitigate them somehow. Well, it's a commercial software, especially if you're a company, it may be very expensive, not if you are academics, and especially not if you are a teaching institution. But in any case, here in Dynamo, we offer the standalone distribution. We compile Dynamo against the MATLAB compiler system, meaning that we need a license in order to write and to compile, but the end users, if they want, they don't need to see MATLAB in their systems in any moment. They can even do scripting with against the Dynamo core without MATLAB. In any case, another thing that may concern some users is the slow execution of MATLAB code. MATLAB is an interpreted language, But in any case, that shouldn't be of much concern because the architecture of Dynamo is thought that all the number crunching happens in C++ and CUDA. And there is a hot transfer of information between MATLAB and C++ and CUDA through MEX files. So that at the end of the day, for users, well, they tend to see Dynamo as a big fat black box. For the developers, I want to underline that, yes, we put under the same roof MATLAB, C++, and CUDA, and inside this black box, there is a different software that we also develop, the MB tools, that has nothing to do with tomography, has nothing to do with subtomogram averaging, is a general toolbox that extends enormously the functionalities of MATLAB for visualization, database management, workflow edition, many things that you normally need when you are developing scientific code. If you want to know more, you can come to one of our workshops. We are trying to go back to normality, so probably in October we are optimistic that in Lausanne we can do a presential Dynamo workshop. Next year we will do another one in Bilbao. In any case, in September there are another virtual workshops. Everybody welcome to attend. They are oriented to hands-on work, so that you will get your GPU station and a list of tasks to resolve in these 2 or 3 days of workshop. And in my last slide, I want to thank the people in the Dynamo team, Stefano and Carlos, with the grant of the National Science Council of Switzerland, Raffaele, working in classification with the grant of the Human Frontier Science Program, and our honorary Dynamo member, Alistair Bart, who is actually working on CCP-EN. And I want to thank all of you for your attention. So if you have questions, I'll be happy to answer now.
Great. Thank you, Daniel. That was an interesting talk. So if anyone has any questions, you can now unmute yourself to ask them. But first, I'll start with some of the ones that came in from the chat during the talk. So Oleg, if you'd like to unmute yourself and ask your question, otherwise I can just read it out for you.
Oh yeah, sure. So first of all, thank you for the inspiring talk. But you clearly explained how the alignment works with fiducials. But what if you have none of them? Do you plan to implement it, or is it already possible? Shall we try to do so?
We have to, we have to implement it because the next step of tomography is going totally cellular, from my point of view. Actually, that is the edge that tomography is contributing in the cryoEM family. We can see cells. So that if you want to do that, the popular option is the cryo-FIB. And when you do FIB milling, you don't have markers. So that I think the markerless alignment is going to take more and more importance in the next. We are working with cryo-FIB data ourselves, and by now what we are doing is what happens if you just take what we have in what you have for fiducial based alignment system, and you throw it. And you throw into it the data that you have from markerless. Because at the end of the day, instead of gold bits, you can go for features in the micrographs. In computer vision, there is a rich literature about features that you can look for, surf, orb, cuts, whatever. So that is what we are trying to do. In principle, we have good results in our experiments, but they are not so easily tunable, meaning that when we put our hands on our TIL series that doesn't have markers, well, we can work for a while, find good parameters for features, then throw it, and then throw our goal-based algorithm on it, It will work to some extent. It won't be perfect, but that's not concerning me that much at this moment, because at the end of the day, what we want to do in the big scheme of things is this feeding back your averages to your tilt series alignment. In other words, what I'm saying is that the idea now is not so much to get a perfect tilt series alignment in one shot, but to get something reasonable in one shot, as automated as possible, and then let the full cycle to do the alignment for you. I'm more interested in automation in this part than in high accuracy alignment.
Thank you.
Great. So on a slightly different topic, there were some questions on like the computational aspects of it. So you talked a bit on how it's set up with MATLAB. There was a question about kind of compatibility with like open source versions of MATLAB, like Octave, for example. I don't know if you've tested this, but does Dynamo run with Octave?
Some of it will work, but I wouldn't advise it. I mean, everything that is based in graphics, Octave has a very different system than MATLAB, so it will not work. For command line operations, it will. I've never done the experiment if I can run a alignment project totally in Octave, but no, I don't advise people to use Octave for Dynamo. If you really don't want to use MATLAB, it's better just to use the standalone.
Yeah, it looks like Pete Meyer has a question as well, so if he wants to unmute.
Yep. Hi. Thank you for the great talk. And I have kind of a naive question about the tomography algorithms you mentioned as far as the feeding back. It seems like there are philosophical similarities to the expectation maximization family of algorithms. Is that something that's illusionary, or is that actually related? In the sense that you're using parameters from one layer and cycling it through another layer and just stacking multiple and cycling.
I would say these two aspects are kind of orthogonal to each other. I mean, they are two different conceptual things. I mean, you can recycle the information from this layer in order to go back to the first layer, and in order to do that, you can use a maximum likelihood setting up, but you don't have to. I mean, I would say that maximum likelihood is a tool, and this cycling the information is a concept. I mean, it's a way. The maximum likelihood is a tool to incarnate this concept, but it's not part of the basic idea.
Thank you. That's why I asked the question. I didn't know the answer. Thanks.
Thanks, Pete. Okay, so one of the other questions we had coming in was, again, you touched on this a little, on the kind of computational requirements and the memory requirements for the system. Are there any kind of broad minimums, you would say, or are there any cases where particle size becomes an issue with either system memory or VRAM on the GPU?
If you go to the Dynamo downloads page and take the Dynamo version that we have right now, I would say it's very difficult for you to find problems with the memory in your GPU, because in this version we don't have yet released our system of feedback between averages and tilt series. It means that the way we deal with things in the GPU is very easy. Basically, you have your template in the GPU, and then you put the particle in the GPU, and some latency goes there, of course. But then you just start moving the template 1,000 times for each orientation. You can have several templates and one particle in the GPU. That's nothing for modern GPUs. So that in principle, that won't be a problem. Next versions, we will need to put full tilt series into the GPU. So that it means that the memory of the GPU will become a limiting factor. Basically, I mean, you will need a memory on your GPU that is enough to have 2 tilt series, which is like, let's say 8. It shouldn't be a problem for commercial GPUs. The more memory you have in your GPU, the more tilt series you can put it for one round, and our system will work faster. But it won't be a real limit, meaning that it is totally impossible to put in, well, not totally, but it will be difficult that each user is able to put 100 tilt series, I mean, the full data set, live in your system of GPUs, so that Dynamo will always, by default, do it in a sequential way. Meaning that the more tilt series you can have in a given time distributed in your GPUs, the faster everything will be, because you will have less steps of loading your data into the GPUs.
Great, thank you. That's all of the questions that we had coming in. So if anyone else has any more questions now, just feel free to unmute yourself. But otherwise, I'd just like to thank Daniel again for a great talk on an interesting piece of software in EN.
Thank you.
And I'll just finish to say that we are taking the next 2 weeks off from the webinar series. So we'll next be back on the 29th of June for Alexandra Bonvin from Utrecht, who'll be talking about Haddock for molecular docking. So make sure to come back for that one.